program, which is for integrating structural biology infrastructures. And in structural biology, we integrate lots of data from different imaging modalities, from different biophysical modalities, very heterogeneous data, very specialized, and also the infrastructures that we use are distributed in many centers across Europe. And I guess that's where the interest came up, why I'm here. So we are also very keen of technologies and on simple solutions, and I want to illustrate that with an example that was not part of my talk, and it's just up here, which I think is this duct tape to isolate a bit this speaker that was having some high frequency before. So simple solutions do occasionally work. So one of the things that we would like to do with integrated structural biology is to follow a basic dogma that actually was first raised in the beginning of the previous century about architecture. And the idea was that form follows function. Like the form, the function, the form of the building was the idea, should serve function. And actually evolution in the cellular level has done the same. So here actually you see like an image that from what I've seen over the last couple of days, most of you would be a bit more familiar with. It's different cells in a slide with the DNA in blue. And for example, here you see like the chromosomes segregated in the mitotic spindle, and here you see another cell in the process of the anaphase with the chromatids being separated. And you see different stages of chromosomal segregation and so on. So for example, what we'd like to do in structural biology is to zoom in such pictures and understand everything in molecular and ideally atomic detail. And for that, for example, we could use like microscopies that would visualize things in much higher resolution. We could use like different fluorescence tricks to visualize certain molecules, but still microscopically. We would like to isolate macromolecules and look at the dynamics of their interaction to start understanding structure in the molecular level. We'd like to look at structures of individual molecules, and of course we would like to look at how molecules, in that case 1, 2, 3 molecules interact. And actually this example shows you just a very, very little detail which you would find here. It shows you how tubulin interacts with the kinetochore, which is in the middle of the chromatid, and this is the interactions that actually are mainly responsible for holding the chromatids and pulling them away during mitosis. And actually this picture is like, would allow even atomic level detail. But ideally, you know, like a concept project would be to start from this cell and go on zooming, as Google Maps is the standard example, and be able to go all the way to that amount of molecular detail wherever it's available. For that, we use a variety of techniques in INSTRUCT that varies from fluorescence microscopies that I showed to you. It goes to X-ray microscopies, which can picture whole cells in high resolution, that's standard light methods. It goes to cellular cryo-electron tomography, which can go to higher resolution than X-ray microscopy, but no longer in intact cells, but slices of cells, so better resolution, but less field of view. It goes to cryo-electron tomography of isolated organelles, that again, it's smaller things, but in yet higher resolution. And we have different techniques that can provide atomic models. That in the low resolution side is small angle scattering. Cryo single particle electron microscopy is undergoing what science has named in 2015 the resolution revolution. It provides now routinely almost for big macromolecular complexes atomic detail at the level of amino acids. I guess most of you are familiar or have heard, actually not familiar, of X-ray crystallography. 90% of the atomic resolution structures currently in the public domain are from X-ray crystallography. This will change in the next 2 decades, but it's still 90%. And nuclear magnetic resonance is also providing for particular problems extremely important information. And all these, we're complementing them with proteomic technologies to show how things interact, and also with biophysical measurements, either on pool of molecules or single molecules, showing again the dynamics of molecular interactions. When you go at this level of information, the real goal of this information, apart from the fact that it increases our basic knowledge, in the translational part allows us to do 2 things. One is to understand drug targets, identify and understand new targets, and I think this is a very important application that it's actually downplayed, and it's downplayed for emphasizing the applicability of these methodologies in drug development, or I should say better, it's mostly involved in the heat to lead cascade, that's where structural biology plays the major role in industry right now. In my, if you prefer, personal view, what sometimes ourselves we underestimate is the contribution of structural cell biology in validating targets that come from this amazing omics technology, especially lately. So INSTRUCT was built to coordinate more than access to facilities. And these are a few things we try to do, and is also to facilitate strategic planning to meet new challenges and within member states and across member states, to coordinate training for acquiring new skills. We want to integrate, and we succeed to integrate with other ESFRI and European Commission projects, coordinate structural biology community, and develop at the end technology in the cutting end. So, INSTRUCT, the idea, the basic mission is still giving access to cutting edge technologies, and for that it needs to operate in 2 levels. On one level, the national funders are the ones that are paying for these amazing infrastructures that we use, all the synchrotron installations, the unfortunately still very expensive electron microscopes, the nuclear magnetic resonance machines. These are things that are we're counting from the tens of millions up to hundreds of millions for such infrastructures. This is national funding for the vast majority of these infrastructures. And what INSTRUCT tries to do, they don't own these infrastructures. These are nationally owned, but they participate in INSTRUCT. And INSTRUCT provides the access process from proposal submission for doing experiments up to review, scheduling, and reporting, and tries to support financially users for going to these infrastructures. The access is internationally open to the members. Right now, there is actually quite strong, not complete European support. There's 12 countries in 15 INSTRUCT centers and around 6,000 members in INSTRUCT. And actually, INSTRUCT has recently been approved as an ERIC, and everything could be signed soon. The founding countries are there. It has been signed, will go on. This in blue is the core countries that will start. The green countries here are associated but have not yet, actually, they were associated in the previous INSTRUCT, they have not signed in the new ESFRI, but we're trying to keep very strong ties with them. INSTRUCT, we think it has been successful. There are lots of publication mentioning there's training courses organized, there's internships given to people that are new to structural biology, and actually we even give small R&D awards for mini projects or to people that are not experts. An issue that perhaps, or at least we hope, might be more relevant to you is this ARIA system, which we have developed actually, it has been developed in the INSTRUCT Hub to give access to research infrastructures administration. So this management tool is not, it has a complicated goal because there are, as I said, 15 centers, and there are thousands of users, and all these users can apply for projects to any of these centers or to any of these instruments, but also very importantly, because we want integrated access, the users can apply to a complicated cascade of these infrastructures. This has to be reviewed, so we need access to a pool of review experts, and all this has to be managed from the INSTRUCT Hub. So we're focusing on the community, access and facility, and also to ideally manage the data that come out. We use, so one big challenge that has been sorted over the last year was a common sign-on. We have integrated different methodologies that allow users, for example, to log into INSTRUCT with their credentials of their local institutional account. And something that ARIA has been involved in is like to go beyond structural biology and support access to other projects, like, for example, Corbel, which is completely, well, it is completely different types of access, to try to create a community that uses a common system to ask for different services, not only, if you prefer, in the area of structural biology, but ideally more in the area of biology and life sciences. A great thing about ARIA is that it can support very complicated workflows. This is not a solid workflow. For example, a scientist can draft a proposal indicating what is required. This is submitted, then we assign a moderator which arranges the peer review in a way which is independent and not dependent on the facility. So you can just not invite your friends only. Then the moderator moderates the review process, ranks them, gives the feedback to the scientist, perhaps leads to the refinement of proposal, then a decision, and then actually even the research and the reporting are managed. But workflows can differ. For example, when the scientist submits the proposal, in some cases, the facilities give feedback to scientists to improve the proposal, and then it goes to peer review. So ARIA, even if some of you at some point want to run your own infrastructures and giving application, if you're involved in it, perhaps you should talk to ARIA for the experience. It allows project overview and what stage every project is. And I'll give you an example of one synergy that I'm much more familiar with. INEXT, it's an EU infrastructure program. We have been funded starting in 2000, at the end of 2015 for 4 years, to give transnational funded access to infrastructure for structural biology. INEXT stands for Integrated NMR Electron Microscopy X-rays for Translation and Research. And our deal in INEXT now was to give access to people that are new, and if you prefer, clueless about structural biology, but they think that this can contribute to their research. We can, once we audit their projects that are suitable for structural analysis, we can take them by hand in different centers and not provide only the facilities, but also the expertise to do a structural biology project. And at the same time, we cater for the established structural biology community, providing high-end access to them. And also, we're funded to improve our infrastructures by doing more research and development. So INSTRUCT is one of our partners. They're managing the access to these infrastructures very successfully. So that's a very good example for how a contemporary project that is funded on a fixed horizon of 4 years is synergizing rather than antagonizing S3s, which sometimes happen. We are together on that. INSTRUCT is providing the administration and the management, allowing INEX to focus on the actual challenges of providing services and developing the services. So for example, this is a quick view. These are the projects that have been accepted for different modality access. As a hint, if any of you might at some point get interested, you see everything that is high-end, there's lots of demand. Anything that is new, because here we're really trying to address new people, perhaps we're not so good in marketing, or it's not interesting, but of course, I think it's very interesting that we try to attract the new people. And please tell your colleagues that might want to look at something with electron microscopy or understand molecular interactions to look at INEX, send their application through the ARIA system in INSTRUCT, and we are ready to provide expertise to them. We're ready to allow them travel to our facilities and help them, yeah, it's there. Anyway, we're not actually doing that bad. You see here the projects, how many days everything is assessed. Within a month from applying maximum, you will know if your project has been approved. Of course, you see that in some things that are new users, this takes about a month. In electron microscopy, because the demand is enormous, this takes a month and a half because the review has to be really a bit extra thorough. In established things, we take 2 weeks, and in other things, we go to 2-3 weeks. It's not bad. We're getting applications all over Europe. We're open to applications also outside Europe. So, getting towards the end, it's a few things about how to manage an international infrastructure and what opportunities this offers. I have to say at this point that I have a bit of bird's-eye view on that, in the sense that I am a basic research scientist, as I hinted, in the Netherlands Cancer Institute. I am the Netherlands representative to the INSTRUCT Council, and now that it becomes an S3, the ministry will go to the council, I'm in the executive, but I am not part of the actual INSTRUCT Hub operations. This bird's-eye view makes me perhaps appreciate how important the central management is, while I'm really not trying to protect my job, because it's not my job, it's, I'm a bit victimized to be here. Oh, it's been fun. What we found is that the external users that came from the facility viewpoint brought new collaborations because they brought new scientific ideas. And for me as a research scientist, that has been the most exciting part. It's not ideas of which project to do, and I offer them a facility so I put my name on a paper. That's not what I'm meaning. People from different fields coming from the facility, very often they gave me ideas about how to do their research, my research. And of course, we hope we contributed to them, and of course, they did also appreciate that widely when they go over the energy barrier to come to our services. So, INSTRUCT and all this team provided the motivation to build the ARIA software, which we think is something that should be looked at. We are now releasing ARIA 2, which has been rewritten from scratch, and it's very flexible, and we hope it's able to support very different infrastructures that need physical or remote access. I think an important feature of ARIA is to facilitate a transparent review process, which is independent and is based on excellence, which is a very big thing. INSTRUCT, a component was that it's now the central hub for developing international training program, either by directly funding it or for enabling better training through making the connections making the advertising, helping in an organization, even if the funding partially sometimes comes from other sources. INSTRUCT is focusing a lot on providing events for networking, but also in participating events like that, in events like this. An issue that we are facing now is that the demanding reporting needs are increasing, both in the national but also in the European context. This comes in this very important concept of accountability. It has a lot to do with access to data, and which is a big concern here. I have to say here, with the data, we're a bit in a dichotomy. The end data of almost anything that comes out of INSTRUCT is inherently accessible in public databases, like the Protein Data Bank. It has EM, NMR structures, everything that we do, the end result is available. The starting data, due to the volume, the actual raw data that leads to these models, which is what most people do, is not so. This is local to the infrastructures, and all the infrastructures have contingency plans. They keep the data, there is transparency, there is accountability, but the sheer volume of most of these data sets has not allowed anyone to seriously tackle the hardware needs that would be needed for all these millions of images that sometimes lead to structures. The needs for that is humongous. There is an effort only for X-rays in Australia, and it's not going that well in my view. Of course, there's a question, do we need it? The answer is that, well, perhaps, I'm not sure, we're certainly not doing it. We're focusing on making available the end result that we're confident that people need, and this is all open, and actually INSTRUCT makes infrastructures available on the assumption or presumption and the need for the end data to be openly available. We have developed good communication procedures because we had to, because it's a distributed infrastructure all over Europe. And one of the things that I think we're good at is that we meet every now and then. And next week we're meeting at Brno in the Czech Republic for the INSTRUCT biannual structural biology meeting. Also, it's a bit late for subscribing. If anybody gets super excited, if you send us an email, I'm sure we might accommodate the 2-3 people if you tell them you're really excited about INSTRUCT. And that was almost the last slide. The instruct.eu site was taken when we started. So we either had to change the website or the name INSTRUCT, we changed the website. Thank you.
Thank you very much. Any questions? So I've got one, and I know that this is probably part of your victimization in the management, but do you, in terms of the process that you put in place to, the operational process you put in place, do you have any goals where you would use like a continuous improvement process to try to attain some efficiencies in how you're operating and decrease some of those times?
So the major challenge there that we're constantly trying to improve is the review process. So the review process, the goal is high quality, maintain high quality by reducing time. So we're putting in place like reviewer ranking systems, expanding the reviewer base, better communication with reviewers, the timing of notifications. Other than that, I have to say that the other operational procedure that needs improvement, and this is a massive challenge, is the initial steps from attracting a user, from the point that you might got interested to INSTRUCT to actually ending up with the correct application. And this is something we should improve ourselves, but it involves a lot of self-criticism and less of feedback, which does not make it easier in my view.